Have you ever wondered how charged particles are being migrated? So this video we are going to talk about that and the technique used and also the principle used on that particle. So we are going to discuss on electrophoresis. What is electrophoresis? Electrophoresis is a laboratory technique that is used to separate charged particles such as RNA, DNA, protein through a gel solution under the influence of an electric current or charge. Then we are going to talk about the principle behind this technique. Number one, we are going to talk about the charge. Molecules move based on their electric charge. Negative charge molecules like DNA move towards the positive electrode. Then positive charge molecules move towards the negative electrode, which is the cathode. Then secondly, we are going to talk about the size and molecular weight. Smaller molecules move faster through the gel, and large molecules move slower because they struggle to pass through the gel porities. Like example, we talk about small children running through a crowded room. So small children run through a crowded room faster than adults. So number 3, shape of the molecules. Molecules with compact or linear shape migrate faster. Complex or bulky shape move slowly. So number 4, we are going to talk about the strength of the electric field. The higher the voltage, faster the movement. But if the voltage is too high, it will cause overheat to the gel or distort the result. Then 5, type of the concentration of the acid. The gel works like a sieve. Sieve is a filter. Agarose gel is used for DNA and RNA synthesis. And polyacrylamide gel is used for protein molecules. And if the gel concentration is high, the smaller pores will lead to slower migration. So number 6, we have the buffer solution. This maintains the pH and provides ions to carry through the electric current, and it keeps molecules in a proper charge state. Then talking about the temperature, high temperature due to current may melt gel or denature the protein. That is why electrophoresis is sometimes done in a cooling system.
Agarose gel electrophoresis is a technique used in the separation of DNA fragments, RNA, and proteins based on their size and charge. The first step for DNA fragment separation is the gel preparation. A suitable amount of agarose powder is put in a bottle and weighed. This bottle can as well be a conical flask. As a low concentration gel, larger molecules tend to dissolve better, while at high concentration gel, smaller molecules tend to separate better. Once the powder has been weighed, a suitable buffer such as TAE or TBE is measured and it is being added into the flask. Since the agarose cannot dissolve until it is heated, the solution is allowed to heat for a few seconds in a microwave. When the agarose has completely dissolved, the solution is cooled in a water bath at about 55 degrees Celsius. While it is still cooling, an electrophoresis tank with negative and positive electrodes is prepared for gel casting. A comb is placed in the tank to create wells for the loading of the samples, and casting dams are also placed at each end in the tank to prevent the solution from flowing out. Once the solution has been cooled to 55 degrees Celsius, ethidium bromide is added to this solution before it is poured into the electrophoresis tank. Ethidium bromide is highly toxic, therefore it has to be handled carefully. Ethidium bromide is an agent of intercalation, thereby intercalating between base pairs of DNA molecules. When it is exposed to UV light, it will fluoresce and DNA bands become highly visible. The more DNA present, the brighter the band. After ethidium bromide is added, the agarose solution is poured into the electrophoresis tank. Then, it is allowed to solidify at room temperature. Once solidified, the comb is removed carefully as well as the casting downs. Next, the gel is submerged in a TAE or TBE running buffer, used to maintain the pH of a relatively constant value as well as provide ions that carry a current. The next step is the preparation of the DNA samples. A loading buffer is added to each sample. This buffer contains glycerol and a dye such as bromophenol blue. It is used to increase the sample's density, allowing it to sink into the gel, provides color, and allow the monitoring of the separation progress simpler. Before loading the samples into the well, DNA ladder, which is a molecular weight size marker commonly used as a standard reference containing DNA fragments of known length, is loaded into the first well, while other wells are loaded with the available DNA samples. After loading the samples into the wells, a lid is used to close the electrophoresis tank. An electric current is applied to pull the sample through the gel. The DNA molecules will travel at different speeds based on their charge and size. The DNA molecules are negatively charged because of the phosphate groups in their sugar phosphate backbone. Therefore, in the presence of electricity, these DNA fragments begin to move through the gel to the positively charged pole. Smaller fragments tend to move faster through the gel than the larger ones. Bromophenol blue moves faster than DNA due to its relatively small molecular size, and by optical control of the moving colored band, the electrophoresis can be stopped before the dye moves out completely from the gel. Once the DNA fragments have been separated, the gel is placed under a UV light. Then, the DNA fragments can be clearly seen as bands. A large number of DNA fragments of the same size is found in each band. The comparison of the DNA bands of the samples to the DNA ladder can help determine their distinguishing sizes.
My name is Mbobombi and I'm talking on polyacrylamide gel electrophoresis page. Polyacrylamide gel electrophoresis is the most widely used analytical method to resolve separate components of a protein mixture based on cells. For these protein molecules of different shapes and sizes, it needs to be denatured. This is done. The protein covered by SDS are negatively charged. When loaded onto a gel matrix and placed in an electric field, the protein will migrate towards the anode, that is a positively charged electrode. They are then separated by a molecular sieving effect based on cells. After visualization by a protein-specific staining technique, the size of a protein can be estimated by comparison of its migration distance with that of a known molecular weight marker. After this, we talk about the procedures. Assembly the glass plate. The first thing we do is to assemble the glass plate on a clean surface. Lay the longer glass plate, that's the one with the spacer down face, and then place the shorter glass plate on top of it. Embed them into the casting frame and clamp them properly, making sure that the bottom ends of the glass plate are properly aligned. Then afterward, place the frame on the casting stand. Casting of the gels. In preparation of the resolving gels, we would require distilled water 4.0 ml, 30% acrylamide mix 3.3 ml, 1.53 pH of 8.8, 2.5 ml, 10% SDS of 0.1 ml, 10% ammonium persulfate of 0.1 ml, 10 mix 0.04 ml, and for the stacking gel, we need distilled water of 5.650 ml, 30% acrylamide mix 1.65 ml, 1 molar trace of pH 8.8, 2.5 ml, 10% SDS of 0.1 ml, 10% of ammonium persulfate of 0.1 ml, 10 mix 0.004 ml. Now prepare the separating gel stroke resolving gel solution by combining all the reagents except the ammonium persulfate, that's the APS and the TME. Add APS and the TME to the monomer solution just before putting it between the glass slide and mix well by swirling gently. Pour the solution until it reaches the mark on the glass slide. If it is okay, you introduce air bubbles. Add a layer of isopropanol or distilled water on top of the gel, which will level the poured gel. Now allow the gel to polymerize for 20 to 30 minutes. Prepare the stacking gel monomer solution by combining all reagents except the APS and the TME. Drain the isopropanol with strips of filter paper and blotting paper. Add the APS and TME to the monomer solution just before pouring it in between the slides and mix well by swirling gently. Place a comb in the stacking gel sandwich and allow it to polymerize for 10 minutes. Preparation of sample. Mix your protein 4:1 with sample buffer and heat your sample at 37 degrees Celsius for 30 minutes in a water bath. Running the gel. To assemble, take out the gel first from the casting frame and clamp them in the gel apparatus, making sure that the short plate always faces inside, and if you have only one gel to run, use a dummy plate that is available to balance. Secure the plate, place them in the cassette and lock it. Afterward, place the cassette in the tank. Fill the inner chamber of the tank with 10x SDS running buffer of pH 8.3. Remove the comb carefully without breaking the well. Now the gel is ready to load the samples. Rinse the loading tip a few times with the buffer, making sure that all the buffer is pipetted out before loading the sample. Insert the loading tip a few minutes from the well bottom and deliver the sample into the well. Rinse the syringe a few times with the buffer after loading it. Attach the power supply by closing lid, making sure that all the connection is correct, that is black and red to red. Set the voltage up to 180 voltage and run for 1 hour. Note, do not allow the dye front to go out of the gel. Staining the gel. After running the apparatus, switch off the power supply and take out the gel plate and remove the gel. Place the gel in staining solution for 30 minutes. Destain the gel until the bands are properly seen. Approximate the molecular weight of the visualized protein bands for comparing them with the molecular weight marker. Cautions: wear your gloves at all times while performing the SDS stage. To ensure proper alignment and casting, the glass plates, spacers, combs, and casting stand gargets must be clean and dry always. Pay special attention while using acrylamide, as it is a neurotoxin.
Sodium dodecyl sulfate polyacrylamide gel electrophoresis, also called SES-PG. It is an electrophoresis method and a very powerful technique in the study of proteins. In SES-PG, proteins are separated in a polyacrylamide gel based on their molecular weight. For sample preparation, loading buffer containing SDS, beta-mercaptoethanol, bromophenol blue, and glycerol is added to the protein sample. Protein samples may be biological derived, for example, from prokaryotic or eukaryotic cell, tissues, viruses, and environmental sample of purified proteins. When the sample buffer has been added, the sample are then heated at 95 degrees Celsius for 5 minutes. Proteins are large biomolecules consisting of one or more long chain of amino acid residue. Amino acids are organic compound that contains amines and carboxylic functional group along with the side chain. Proteins are formed by linking amino acid with peptide bond and fold into one or more specific spatial conformations, driven by a number of interactions such as hydrogen bond, hydrophobic interaction, disulfide bond, and ionic bonds. SDS is an anionic surfactant and contains a polar head group with a net negative charge at the end of a long hydrophobic carbon chain. SDS denatures the native proteins by disturbing the hydrogen bond, hydrophobic and ionic interaction, while the reducing agent beta-mercaptoethanol is used to cleave to the disulfide bonds. Also, when a protein mixture is heated, the SDS binds firmly to the protein, and the intrinsic charges of this protein become negligible when compared to the negative charges contributed by the SDS. This treatment brings the folded proteins down to linear molecules. With non-negative charge, therefore, these proteins can be separated in a polyacrylamide gel based on their molecular weight. For a gel preparation, a separating solution with pH 8.8 and a stacking gel solution with pH 6.8 are used, and both consist of acrylamide, bisacrylamide, ammonium persulfate, and timide. The gel is produced by polymerization between two glass plates anchored vertically in a cassette. The separating gel is poured first. The polymerization reaction is a vinyl addition polymerization initiated by free radical generating system. Ammonium persulfate is used as a free radical initiator, while timide accelerates the rate of formation of free radicals from sulfate. These free radicals convert acrylamide monomers to free radicals, which react with unactivated monomers to begin the polymerization chain reaction. The elongating polymer chain are randomly cross-linked by bisacrylamide, resulting with a characteristic porosity. The size of the gel is related to the ratio of acrylamide to bisacrylamide. Low concentration is preferred for high molecular weight sample. The stacking gel solution is poured on top of the solid separating gel, and the plastic comb is placed on the top of the stacking gel during polymerization, enabling the formation of small wells in the gel. Once the gel is completely polymerized, the comb is removed and the gel cassette is placed vertically between two electrodes. Positive electrode located at the bottom of the gel, whereas negative electrode positioned at the top of the gel. The gel is inserted into a chamber and a trace glycine chloride buffer system with a pH of 8.3 is produced to allow the conduction of electricity through the gel. SES is also present in the gel and running buffer to make sure that once the proteins are denatured, they stay that way throughout the run. For sample application, in addition to samples, a molecular weight size marker is usually loaded into the gel. This consists of protein of known sizes and thereby allow the estimation of the size protein in the actual sample, which migrates in parallel in different tracks of the gel. Each sample is pivoted into its own well in the gel, and as we have seen previously, bromophenol blue and glycerol are present in the sample. Bromophenol blue is a dye that is used for visualizing the sample in the well, and glycerol increases the density of a sample and is used to make the sample to the bottom of the sample well rather than just float out and mix with the running buffer. After the sample application procedure, an electric field is applied across the gel, causing the negatively charged proteins to migrate across the gel away from the negative electrode and towards the positive electrode. Also, small proteins migrate relatively easily through the mesh of the gel, while larger proteins are more likely to be retained and thereby migrate more slower through the gel. The stacking gel is used to ensure that all the proteins arrive at the separating gel at the same time, because the gel wells are around 1 cm deep. So, in the absence of the stacking gel, the protein will all enter the separating gel at different times, resulting in very smeared bands. As we have seen previously, the running buffer consists of glycine and chloride ions. So in the sample well, we will have protein, chloride ion, and glycine. Protein and chloride ion are negatively charged, while glycine exists in 3 different charge states depending on the pH of the isoelectric pointer. pHi of glycine is equal to 5.97 at this pH. Glycine carries neutral charge due to the gain and the loss of proton at pH below its pHi. Glycine carries a net positive charge at a pHi above its pHi. It becomes negatively charged. The running buffer pH is equal to 8.3, which above the pHi of glycine. So at this pH, the glycine is negatively charged once the electric current is applied. Proteins, chloride ions, and glycine are forced to enter the stacking gel, where the pH is equal to 6.8. At this pH, glycine switches predominantly to the material neutrally charged state. This loss of charge causes them to move very slowly in the electric field. The chloride ions, on the other hand, move most quickly. Chloride migrates in front of the protein as leading ion, and glycine molecules migrate behind the protein as initial Consequently, proteins are trapped and concentrated into a narrow zone between the chloride ion and glycine. This procession carries on until it hits the separating gel, where the pH switches from to 28.8. At this pH, the glycine molecules are mostly negatively charged and can migrate much faster than the proteins. Proteins are separated, so higher molecule weight proteins move more slowly through the porous acrylamide gel than lower molecular weight proteins due to the relatively small molecule size of bromophenol blue. It migrates faster than proteins, and by optical control of the negatively charged migrating colored bind. The electrophoresis can be stopped before they die and before the sample have completely migrated through the gel, and leave it at the end of the electrophoresis separation. All proteins are sorted by size and can be analyzed by the other method, for example, protein staining method and immunological detection, such as the Western blot technique after removing the gel glass plate. The stacking gel is disconnected and the separating gel can be stained. Chromacy blue is most popular protein stain. It is an anionic dye which non-specifically builds the protein. It detects blue band in a separate gel to determine the unknown mass of biomolecule by the distance traveled in the marker.